Hi, how are you doing today? Well, I hope today is part three of my three-part video series. If you haven't seen the other two, then you need to play catch up because we are studying hello, thanks for joining. We are studying the microbial growth. First video, we talked about temperature and how that affects the growth. Yesterday, we talked about the pH and how that affects growth. And today, we are going to talk about osmotic pressure. Before we get started, I will introduce myself. My name is Rochelle, and I am your microbiology study strategist. Hello. Microbiology study strategist. I make it my mission to position you to understand, retain, and remember microbiology. That is the goal. So I covered what we did, and you know who I am. So let's get started with the lesson. All right, let me flip this around. All right, looks good. So put my glasses on because I can't see. I'm blind. So today we are talking about osmotic pressure. First up we have isotonic. And of course it's not working. Okay, there we go. Isotonic. And let's go with hi, how are you? Thanks for joining. Blue. Let's go with blue for isotonic. So isotonic. And this one is isotonic. So what we see here is equilibrium. There is the same amount of water or solvent and solute versus inside and outside of the cell. So there is equilibrium. The next one is hypotonic. And this one here, you see there is a high concentration of solute versus outside of the cell. And then hypertonic. We see more of the solute outside of the cell versus inside of the cell. That's basically what it is. So what you need to do is actually remember this. So what I do is use the letters within the, let's move that a little bit. Use the letters within the name, within the word to help me remember what the definition or meaning of that word is. So what I will do is use the S here in isotonic, and I will spell same. So A-M-E. Because the equilibrium, the equilibrium between the concentration inside and outside is the same. So I would use the same. For hypotonic, I would use the O, and I would spell out osmolysis. Now, osmolysis is the destruction of the cell. So here we see a high concentration of solute. That means water will have to come inside. Water has to come inside the cell. And if the water comes inside or goes inside the cell, then that means that the cell will swell and break. So this is lysis. That's what it means to break. Think of a water balloon. An example here. An example is a water balloon. You pour water, you have water going into that water balloon, and it'll expand and expand. If you put too much water, that balloon will break. This is osmolysis. So think of it that way. The next one is hypertonic. Well, let's go back to hypotonic. So we have osmolysis. For an example, think of a water balloon to explain it. And then we are going to use the I here for in, because water is going inside. Water going inside. So water going in. So we're using the I here for in, water going in, and that's how you can remember hypotonic. Then on to hypertonic, we are going to use the P in hypertonic for plasmolysis. Again, we see this lysis, meaning to break, so you know something's going to break. The cell is going to break. So in plasmolysis, we see there is a high concentration in hypertonic, a high concentration of solute outside of the cell, which means that water has to leave the cell to reach equilibrium. So water is leaving the cell. If the water leaves the cell, then the plasma membrane will then shrink. And then if there is a lack of water, then there is a lack of nutrients, and the cell will break due to the shrinkage. Again, a great example is a water balloon. So now that you have a full water balloon, then you would pour out that water, you'll squeeze out that water, you'll get a shriveled up balloon, a balloon without water. So that is an example that you can use. How would you remember? How would you remember this? So you're using the P for plasmolysis, water leaving, you're going to use the O for out. So water is leaving, or water is going out of the cell. So that's the study portion. Isotonic, same. Hypotonic, O. We're using the O in hypotonic for osmolysis. We're using the I for in because we know that water is going inside of the cell. Hypertonic is, we're using the P for plasmolysis, and then O for out because water is leaving, water is going out of the cell. The main goal is to reach equilibrium. That is the goal. Next, we are going to get some examples. So we're going back to hypertonic. Let me move this up some, just a little bit. So hypertonic, we see this in preserving food. So that is a great example, like salted fish, or making honey, or jam, making strawberry preserves or apricot preserves. That is, those are examples of preserving food using a high concentration of salt or a high concentration of sugar to preserve the food. This will then you will see this action, plasmolysis, occur where the water will leave out of the cell and enter into the environment that it's in, and then causing the cell to die. And since there are dead cells, then they cannot, and when you ingest the food, it cannot harm you because they're dead. So that's how food is preserved. What else do I want to say about this? Where can you find them? You can also find them in salted, highly salted environments like the Dead Sea. A Dead Sea is a place that you can find hypertonic microbes that can live in high concentrated salt environment, and they are extremophiles. They are called, not extremophiles, extreme haliophiles. Extreme haliophiles. halophiles. And they can grow in areas like the Dead Sea. And I think that's all. An example for hypotonic. Hold on, one more. Another example for hypotonic, we have distilled water, where the minerals, the salts, are taken out of the water. So the environment has very low amount concentration of salts or minerals outside of the in the water, and then this will cause cells that could be in the water to die through osmolysis. I think that is it. That is the teaching study. I gave you 3 study tips to help you remember by using the letters in the word. Now we're going to go on to the questions, which I have from the previous day. That was yesterday's teaching. Oh, crap. Go back. Yesterday's teaching right here about the pH. If you missed it, you can go back and catch it. So here, let's see. There. These are the questions that you can answer and put it in the comments. Question number 1. What type of microbes are able to tolerate acidic environment? And we talked about that yesterday. What is the optimal temperature of pathogenic bacteria? We talked about that a couple of days ago. And today, which organism can live in hypertonic environment? That was what we covered today. And you can put your answers in the comments. Let me flip this around so you can see me. All right. So those are the questions. That is the topic, the teaching, the studying, the testing your knowledge. If you found that this was helpful for you, feel free to click the link in my bio, because this is what I do in my study sessions, my Sunday sessions, where I teach you the topic and we study. I give you tips on how to study, how to retain it, and then I test your knowledge with questions and worksheets so that you don't leave the session not knowing the material. I'll take these off too. You're not leaving the session not knowing what you just learned or studied. Like in lecture, the professor can go on for a whole hour and you can leave and not know anything he or she taught you. But I wanted to fill in the gap, and that's what I do. I teach, study with you, and then test your knowledge to make sure that you really understand the material. So if you find that this is helpful for you, again, click the link in my bio and sign up for my Sunday sessions. If you don't need help in understanding the material, you can understand it, but the structure of how to study is something that you can't grasp. Then you can click my Book Now button on my Instagram page, where I create for you 4 weeks of study, personalized study plans for you. So you'll go into each study time knowing when you will study, what you will study, and how you will be studying microbiology. And that is it. Again, if you have any questions, you can put in the comments. If you like, you can answer those questions that I presented and put in the comments. Or if you have any questions, you can also DM me if it's personal. Oh no, hold on, not personal. If you're too shy to put it in the comments, you can put it in, you can DM me. And I think that's about it. My name is Rochelle, and I am your microbiology study strategist. And I will see you, not tomorrow, because today was the last day. I'll see you next week. How about that? All right. Have a good one.